Hello.
G'day, how you going?
Not bad. Pleased to have you with us. None of this will make the final cut.
Oh, no. None of that hypnotist stuff?
That might be a separate pickup sheet. Whole film. How did you get into science? What was the attraction? Why? Just walk me through how you ended up here.
Oh, okay. All right. This could be a really long story. So I actually have wanted to be a scientist, more specifically an immunologist, since I was 11 years old. I grew up, so we were refugees, we were boat people. My dad was a doctor back in Vietnam, and when he came back, so when we came over here, he had to go back to med school again, and we grew up in a household where my mum and dad would tell stories about people like Marie Curie or Albert Schweitzer, who really changed the world. And so I kind of like grew up thinking that's the sort of thing that I would like to do too. And you know, I spent a lot of my days, early days anyway, in school, hiding in the library at lunchtime, reading Scientific Americans, and that's when I first read about this amazing Australian immunologist named Macfarlane Burnet, and some of his theories and some of his ideas. And I was sold. Since 11, I've always wanted to be an immunologist.
Okay, I don't know anything about this gentleman Burnett, so you need to. What was exciting about him? Teach me, my lord.
Right. So I think one of the things that's really always struck me was how does anything work? How does the body work? How does, and in particular, how does our body know how to tell the difference between something that's dangerous to us, like a virus or a bacteria or some sort of parasite, and how does it tell the difference between something that's dangerous and something that's harmless, like the foods that we eat? So that ability to discriminate between, you know, potential danger and something that, you know, is mostly harmless, that's kind of like really fundamental to what the immune system does. And when that falls apart, well, several things happen. One of which is we develop infectious diseases because it doesn't recognize the threat posed by these pathogens. Or we develop autoimmune disease when it misrecognizes a part of our own body as a danger and starts attacking ourselves.
They're the two extremes.
They're the extremes. Or we develop allergic diseases because something that should be harmless, like, you know, a protein that's in a peanut, our immune system thinks is a terrible threat and we must eliminate at all costs, even at the cost of posing a threat to the life of the individual involved. So these are really basic fundamental questions, and what was really exciting was that what I was reading was, as a child growing up, was that there was a theory that might be able to explain how these things might work. And one of the most exciting things was that this theory could be tested. You could do experiments. You could go out and see whether this theory was correct or not. And I think that's fundamentally the difference between something like science and belief systems. You know, science is all about falsifying, disproving things, testing things, and seeing if things hold up and stand, you know, the test of time. And then if it's correct, then you'll be able to predict what the consequences might be. And that deep knowledge then also then allows you to then get in there and change some of the switches, some of the variables that then might actually change the outcome. So someone who, say, if they were exposed to a peanut allergen, may be, you know, their trajectory might be, okay, this person's going to become allergic to peanuts and, you know, at risk of dying if they, you know, accidentally eat a nut-containing food. Can we get in there and, you know, tweak the levers so that the immune system rewires and resets itself and say, oh, actually, this is not the threat that we think it is, and change the course of that individual's life? Or more importantly, I think fundamentally one of the other really interesting things about what Macfarlane Burnet's theory implied was that actually that ability to discriminate self from non-self, what's you and what's the outside world, that's what's gone awry in cancers as well, because fundamentally a cancer is a part of yourself that's changed in some way. It's a cell that now no longer follows the rule of social behavior. And its antisocial behavior, for want of a better term, is not being recognized and being policed by the immune system.
It's the drunk at the party, isn't it?
The drunk uncle that you don't want to know, right? So this idea then suggests that maybe if we understand how the immune system interacts with the cancer, then and understand what's gone awry, so that the immune system now, for whatever reasons, decided, okay, you know what, a bit out of control, but we'll tolerate this behavior. Can we change it so that that policing becomes more effective? That the immune system says, actually, we're not going to tolerate this. We're going to get rid of these cells because, you know, they're bad news for the host, for the person involved. And so that's, you know, I guess part also where our interest in cancer immunology lies. Am I rambling too much?
Just enough.
Okay, all right.
And you're not rambling, it's actually really interesting. but that was more about discovering what Burnett, how Burnett inspired you and what, because that set the fire under you. I was reading Lord of the Rings, you were reading about Burnett, so it's like, what was the excitement there? It was just the ability to change people's lives?
Yeah.
That's a bit there. So you left high school, so you majored in...
So when I was in high school, I wanted to do, you know, I wanted to be an immunologist. So my teachers were horrified because they didn't want me to do science. They got in a couple of professors from the university who convinced me that actually what I wanted to do was not just research, but biomedical research. And to be more effective, they suggested I first do medicine. So I actually first trained as a doctor, as a medical specialist in clinical immunology. So so after high school, I went to uni, studied medicine, and then I trained as both a pathologist and a physician. So I did a double fellowship. And then it was only then that I did my PhD and started my scientific career.
Which uni was this? Where'd you go to school? Where'd you go to uni?
So I went to school down the road at Sydney Boys,
Okay.
and from there I went to Sydney University, and I did my medical training at the Royal Prince Alfred Hospital in Camperdown.
No overseas trips for you?
So I did my PhD then at a place called the Centenary Institute at Sydney University, and so during my PhD, so during my studies, it became apparent that, you know, one of the key cells in the immune system is this cell called a B cell, because that's the cell that turns into a plasma cell that makes antibodies.
Walk me through that, I'm sorry.
Sorry?
Walk me through that a bit more.
All right. So,
and I'll clear my throat.
okay. So one of the ways that the body deals with threats is to make antibodies. These are proteins that can specifically recognize the virus or, you know, the danger and eliminate them. And these antibodies are made by plasma cells, which are the products or the, you know, plasma cells come from B cells, right? So we were really interested in B cells and how B cells learn to make antibodies. And so to be able to do that, you need to be able to track them. And so for my PhD, we developed a model that allowed us to track the B cells by engineering the receptor on the B cells that allows it to specifically recognize these molecules or proteins on the surface of, you know, potentially pathogens, right? But it was one, so each B cell can recognize one specificity, right? So that was what Macfarlane Burnett was famous for, the clonal selection theory, which is the foundation of immunology.
So it's like targeting? It's like
It allows it to specifically target. It allows it to recognize and target, and also allows it to remember, because, you know, the virus, like if you think about something like SARS-CoV-2, the spike protein on the virus, that's very specific to SARS-CoV-2. We have B cells that recognize just that spike protein, right? Now, it makes antibodies against that spike protein so that when the virus comes back again, that's recognition allows it to make that antibody again. So that those are like the key aspects of the immune system, that ability to recognize and to remember, right? That's the sort of that makes the immune system probably one of the most powerful things that we have at our disposal, actually, to fight cancer, because the ability to recognize a cancer cell and to remember means that we could potentially, if we can learn how to exploit those capabilities, we can teach the immune system to recognize and eliminate the cancer, and not just eliminate the cancer there and then, but remember, so that if the cancer comes back in any way, shape, or form, it might be able to then also target it and destroy it the next time round. So what was I talking about, rambling about?
Well, no, we were going back to what inspired you, then we went to what school you went and
and did I go overseas? Yeah, so during my PhD, we developed this model that allowed us to track B cells that could target this specific protein. And around that time, people had just started doing something called intravital microscopy to track the immune cells, because the problem, of course, is that for a B cell to become a plasma cell, it's an enormous...
Okay, let's go back a bit, because I'm loving what you're doing, but how long ago was this?
A long time.
No, it's not. Not from my point of view.
This is about 25 years ago, when I was doing my studies.
Year 2000, around 2000.
Yeah, 2000 I started my PhD. And then in 2006, I went to San Francisco to do my postdoc at the University of California in San Francisco. And what we were doing there was actually using this incredibly exciting new technology called intravital microscopy, which allows us to get into the living tissue inside a living animal to see these immune cells in action. And this is really important, because if you think about it, for a B cell to become a plasma cell, that's an incredibly, for the B cell to become a plasma cell, it has to change so many things about itself. It basically has to transform itself from a nice, quiet, resting B cell to this enormous antibody-producing factory, this cell that's just pumping out huge quantities of protein every day, the antibodies that help us fight the infection. So for the cell to make that decision, we really need to be able to understand those decision-making process, and that's the decision that's made inside the living body, inside these structures called lymph glands. So if you think about it, when you have a vaccine in your arm, you get swelling and pain in your armpits. That pain and swelling are your lymph nodes, your lymph glands swelling up in response to the vaccine. And that's what we need to do, get into those lymph glands and see exactly what's happening in a live animal. And that was what intravital microscopy allowed us to do. And so that was the technology that was only just coming online, and we were really excited about it. And so we packed our bags and set off to San Francisco to learn how to build these microscopes and how to make them work.
But this is exciting. And how new was that technology in 2000? It was like...
Sorry, was that I went in 2006, sorry, yeah. So the first papers had only just been published 1 or 2 years earlier. And the lab I went to was one of the very first labs to have adopted this technology. So it was very much in the early days. And of course, at the time, there were, we had to build our own microscope. There was no company that was making microscopes that you can just buy off the shelf and put in your lab and run the experiment. We had to buy all the component parts and assemble them and work out how to fit it all together.
God, who did the theory on that? Who knew enough about optics? Was it electric microscope or optical? Or?
So it's a two-photon microscope. So two-photon microscopy relies on, so it's a fluorescence microscope, right? So essentially we're making cells glow in the dark inside these animals, and so it's about making the invisible visible and being able to see through things. And so normally with light microscopes, you're dependent on visible light to excite the fluorophore, which then emits fluorescence light, which then comes back at you, right? Now, the problem, of course, is that if you look at the skin, you can't see through the skin. And the reason why you can't see through the skin is because of a couple of things. Some of the light gets reflected back, but also the light can't penetrate through the skin because it gets absorbed, for example, by melanin, by lipids, pigments, and so forth. Now, a lot of that is light in the visible range. With two-photon microscopy, we use a near-infrared laser to excite the tissue. And so once you move outside the visible wavelength into the longer near-infrared wavelengths, light is able to penetrate deeper through the tissues. There's less scattering of the light. And so that allows us to then do deep tissue imaging without having to take the gland out of the body and then cut sections. So normally when people do microscopy, they think of, taking a slice of the tissue, putting it on a slide, and then putting the slide under the microscope. Instead, what we're doing is we're using this laser to take, we're basically using light to cut sections through a living lymph node inside a living animal.
Wow. And that's exciting.
It's pretty cool.
You just put me in mind of you. Is it similar to the microscope they've been using on the recovered library in Ukrainian or Pompeii?
So
They managed to go in and, I'm sure you know it, but each level, so they can actually read this folded up papyrus book without opening it.
yeah, so the concept is very similar, right? Where by using longer wavelengths, you can penetrate through tissues, right? And by being able to penetrate through tissues, you're then scanning what we call a Z-stack, or an optical section. And then you can then reconstruct a three-dimensional image of the tissue. Now, what's really exciting is that because we're using longer wavelength light, and it's two-photon, there's less tissue damage by the light. And that allows us then to take sections repeatedly. So if you take a section and then 30 seconds later you take another section, over time you might take hundreds of these sections. You could then stitch together a movie and you can see the pattern, the behavior of the cells. You're now not only looking at static images in 2 dimension, you're looking at dynamic interactions between cells, between the immune cells, between the cancer cells in 4 dimensions. So that provides us with an unprecedented ability to then now start asking, what are these cells doing and why are they doing it?
It's essentially time-lapse photography, is it?
Yeah, it's making movies.
Some people don't know what we do. Excuse me, my throat's going. Wow. Okay, so.
And so I guess the other technical aspect of it is that we can actually, with the scanners, we can actually image at incredibly fast frame rates, to achieve what you would call video rate time-lapse imaging. So and what we would call real-time intravital microscopy. So the microscope allows us to then dissect these processes at quite incredible spatial resolution in terms of the pixel size how big the objects are that we can discriminate, so we can get down to subcellular resolution. And then also in terms of temporal resolution, the time steps in between each image.
What we would call persistence of vision.
Yeah.
Yeah. Okay. Just checking.
Yeah.
Yes. Oh, thank you.
Oh,
Do you want a drink?
no, I'm good.
Obviously, somebody didn't like my coffee. Okay, so you left university, you did your PhD, and then what?
Well,
How?
after my
Well,
postdoc.
then how? Yes.
So you know, I mean, the postdoc was just amazing because it really opened my mind to the possibilities of what you can do, and I think one of the great things I learned from my postdoc was that quite often, a lot of times, there have been these really difficult, hard to answer questions in the clinic, in the lab, difficult, hard to answer questions that actually, maybe when we think about it, are really difficult, hard to answer because the technology didn't exist for us to answer these questions. And what my postdoc taught me was actually, if the technology doesn't exist, what's stopping you from inventing the technology? If the right microscope doesn't exist for you to see what you really want to see, to make the invisible visible, then what's stopping you from doing that? And I think what's really been really exciting was that really opened up to me the idea that actually, you know, throughout my career, especially with my medical training and then my specialist training and then my PhD and my postdoc, what I was doing, was really funneling my efforts into narrower and narrower specializations. But actually, the revelation was that makes me really, really knowledgeable and expert in one particular area, but I really need to have expertise at that depth in multiple areas, not just biology, not just immunology, but physics, optics, chemistry, maths. And it became apparent that actually I can't do all those things. And the obvious solution then is to then connect with people who are the physicists, who can invent the microscopes, the mathematicians who can solve, the equations that tell us how these cells are behaving, and so forth. And so it became really clear then that the key to what I was going to do moving forward was going to be an interdisciplinary approach where, we have a critical mass of domain experts who can work together to solve these insoluble problems. And I think, that's probably one of the driving forces for when I came back here, came back to Australia, to the Garvan Institute, and then ultimately to set up my own lab. The motivation was, how do we engineer a situation where we have all these capabilities at our disposal, and how do we resource something like that? Because that's not everyone talks about doing it, but to actually do it is not something that's, very easy to achieve.
Get it? So you came here, you joined, you put the team together, basically, did you? Because you do make me think that 17th century and 18th century doctors who were like, whether it was Darwin or Benjamin Franklin, said, okay, it's just me, and I became masters of 6 or 7 different skills,
Right.
And that became less and less the thing to do in modern
No,
science.
absolutely, yeah.
You just concentrate on this. I remember meeting one professor who said, I said, what do you do? Medieval historian.
Mm-hmm.
I concentrate on nuns in Britain between 850 and 950. Good. So I realised that's...
Don't ask me about 951.
Yes, exactly. That's beyond my area of expertise. Is that... It seems you're not doing that. I'd like to hear about the comparison between that, in your words, not mine, between old-fashioned and new-fashioned, and the fact that you are looking for a broader scientific basis within the team.
Yeah. So I think the key to what we do, and I think increasingly the key to what a lot of labs do in trying to push back, the boundaries, is to really to try to get as much dialogue as possible between different ways of thinking, different approaches to problems. And yeah, so for us, that diversity in opinions, in backgrounds, those things really shape how we approach a problem. And having an environment in which people are really comfortable to, discuss their very, perhaps, out of the box ways of thinking is really key, because if you think about it, everyone's trying to solve the same problem, but everyone's bashing their head against that wall in the same way. We're not going to add to the solution by also getting in line and bashing our head against the wall. So our approach is then to go, okay, is there another way of doing it? And can we imagine a different way of doing it? And if that different way doesn't exist, can we invent it and then pursue it? And increasingly, I think you'll find that, even if you look at the published literature, the big papers in the big journals, going back several decades, they may have one or two authors. Nowadays, it's not uncommon for them to have, 50 authors. And I think that's not a bad thing, because, that interactive, collaborative nature, I think, is often going to be much more successful, because you have then at your disposal all these amazing resources, which are mostly human resources.
It's complementary, isn't it? Many people being complementary to each other, more like complementary nuts.
They just make you thirsty.
I'll tell you the joke later. I'm sure you know it. Going back to when you were a kid too, the inspirations. When I'm going back through, I'm going to have to cover the history of medicine. So,
Oh God, okay.
Yeah, from cowpox to smallpox to penicillin to Hippocrates to Galen, and cutting dog animals open and all that. But we'll manage to do that. Was there anything from the past, you mentioned the Curies, that inspired you, or said, wow, that's so cool? And if not, fine.
I think in my clinical experience, I've had a few examples where I've just went, wow. I think probably one of my favorite stories is when I was training at the Royal Prince Alfred Hospital. There was a respiratory physician there named Peter Bai, and he was an expert in cystic fibrosis. And he had this one patient. So cystic fibrosis is a terrible disease. It, affects a channel that's really important in the function of so many different organs. But, patients get terrible lung disease. They also get terrible diabetes. Their pancreas doesn't work, etc. But he had this one patient who had terrible diabetes, and you would imagine should have terrible lung disease as well, because the kid's pancreas just didn't work. But his lungs was pretty good, and Peter noticed that actually his lungs were better in the summer months than the winter months. And that kind of makes sense anyway, because typically cystic fibrosis, you can get chest infections, which makes your disease worse. You're more likely to get chest infection in the winter months. But what Peter did that I thought was quite extraordinary was he sat this young fellow down and said to him, why do you think your lungs are better in summer than winter? And he said to him, well, I surf more in summer. And then Peter recognized and realized that maybe when he's surfing, he's exposed more to the aerosols from the salt spray. And maybe the salt, that aerosolized salt, is actually helping with his lungs. And so then he did a study. He did a trial testing aerosolized saline. and compared it against this super expensive drug made by a drug company that was meant to help clear the mucus in these patients, and it performed better. And so the inspiration to me from that story was that actually we can learn a lot from our patients and what they tell us. The information's all out there. We're just ignoring it because we're not looking or listening in the right places.
Good story. And negative ions.
Well, I think the saline, so one of the problems with cystic fibrosis is it's a chloride channel, right? And so they have problems with the thickness, the viscosity of their mucus, so then they can't clear it, so then it collects. And so the salt water, essentially, the aerosols in it, helps them improve the viscosity, and then it helps the clearance of it. But yeah, the idea that actually the patients themselves have the solution.
That's very old-fashioned.
I know. I'm an old-fashioned guy.
Moments of, have you had any eureka moments in your life, or where you think, this is the coolest thing I've done?
So the problem is I'm like a goldfish. Like pretty much the last thing I see is the coolest thing I've ever seen. So yeah, but I do think it's an enormous privilege what we do, because with I kind of think like what we do is the microscopic equivalent of David Attenborough, because we invest an enormous amount of time and effort to make these cells visible and to be able to see what these cells are doing in the dark, kind of like, spending months setting up a hide just to capture that 5 seconds when some exotic bird does some exotic dance, and that's it, never to be seen again. And so I feel enormously privileged, because when we do an experiment, we are the first to see what we are seeing. And so even if it's not, like some amazing discovery, even though a lot of it is, okay, that's cool, what does it mean? We don't know quite yet. I still get super excited, because it's kind of like, wow, no one's seen this before. We're seeing it for the very first time, and it's now up to us to work out what it means. So that I think is really exciting, and just about every experiment, that's kind of like what we're doing. So,
do you think there's such a word as failure in research?
I think research is all about failure. I mean, and here I feel very strongly. Science is about disproving things, and failing is really important, because you have an idea, a hypothesis, you go out and you test it, you show that it's wrong, you refine that, you adapt it and you change it in face of the data that tells you that that was wrong. And that's how we slowly get to the point where we know more about a system, like the immune system, like how the immune system recognizes and deals with cancer. And that knowledge then allows us to then create maybe tools or drugs that allows us to then target, and treat those conditions. So to me, failure is really important. Science doesn't work without failure. And I think the idea that, you have to be right every time, I think is probably something that would pretty much, I think, not be consistent with good science. I remember growing up in the early days when CT scans weren't that widely available. One of the things that I was taught was that a good surgeon would have taken out at least one normal appendix in their career, in their surgical lifetime. And that speaks to the idea that, you got to get it wrong. If you're going to get it wrong, you're going to get it wrong or on the safe side. And I think that's really true as well in science. I think a really good scientist would have been wrong most of their career. And that's how they've worked, arrived at being right at the end.
I think you've answered it, but I have to ask you again, what do you love about your job?
So there are so many things I really like about what I do. And I think increasingly I recognize that as I get older, the things that I like have changed a bit. It used to be when I started, the excitement of doing an experiment, seeing something for the very first time, and thinking, I did that, like I made that possible, was super exciting. Increasingly now, I'm really thrilled by the idea that actually there are young people coming through the lab and they are able to have that same experience. To me, that is much more thrilling because increasingly we are seeing some super bright people come through the lab, and sometimes I look at them and I think, one day you're going to do something spectacular, and I'll be able to say, I had a small role to play in you arriving at that point in your life. And I think that will be really exciting.
Which begs the question, how far have we come, just to give us a heads up in 20 years, the changes that have made? I know they're remarkable, and one of you could talk a little bit about them.
So I think this is one of the things, right, isn't it? On the surface, it looks like there's a lot of activity and we've come a long way, we've learned so much. I think definitely, for example, we now have cancer immunotherapy as an additional pillar of cancer treatment, and to me, that's super exciting because the immunology provides us with the opportunity to now cure cancers that were previously untreatable, but now we're thinking might be curable. So we have come a long way in many ways, but at the same time, there are so many things we still don't know. And it's one of those things, the more we learn, the less we realize we know. And I think that's great, right? Because, you sort of like travel along that curve of total ignorance, thinking you know everything, to learning a little bit and going, okay, actually, now that I've learned a little bit, I realize there's so much more for me to learn. And and increasingly also, what's exciting is the recognition that actually a lot of what we don't know, the answers might already be out there if we just look and listen in the right places.
How has the Australian Cancer Research Foundation helped, or has it helped?
Yeah, I would say that one of the really challenging things for medical research is that, and particularly for the sort of research that we want to do, is that it is quite high risk, and quite often to do what we want to do requires an enormous leap of faith.
What do you mean by high risk?
So when, okay, maybe I'll backtrack. When there's a problem that you want to solve, you can either sort of do what everyone's done, and that's safe, and it might, which is, for example, you might be able to chip away at the problem in some small way. And that's important, that's good, that's work that needs to be done. But the other way to look at it is go, okay, that's going to take an enormously long time and effort and energy. Is there another way that we can do it that might, not give us an answer, might not give us a reward, but which, if it plays out, will give us a reward that is much bigger and much sooner than that other way. And so that's kind of like high risk, I guess, and it's unfortunately the sort of science that doesn't get funded in Australia because it's high risk and the environment is very conservative in terms of the funding landscape. But more importantly, that approach requires an enormous amount of investment in infrastructure, in equipment, in things in a space in which I think the Australian Cancer Research Foundation is unique, and in many ways the entire Australian research community is dependent on it for now, because they do see potential, they do see the fact that these investments need to be done, and that these equipment need to be made available for all researchers in the community. So the approach that I mentioned to you before, where we need to work with all these different disciplines to get together and solve this problem, that's the sort of thing that ACRF is very good at facilitating.
What have they done for you? And this is an honest question, I'm not sure. I know they've given money, but I'm not sure in what aspect.
So
I recognise it's not just you, it's the team, if you like.
yeah, absolutely. Yeah, so for us at the Garvan Institute and to, with all our partners around Australia and internationally, ACRF has been a godsend because what we needed to do, not just wanted but needed to do, was really take our intravital imaging to the next level. We didn't want to just scratch the surface because, what we really wanted to do was to be able to get really, really deep into these tissues, deep into these dark spaces that people hadn't been before. And that needed a new generation of microscopes and a new approach.
Oh, I'm so sorry.
That's all right.
Oh. Yeah. Hang on. No, it should have come. A new generation of microscopes is when I screwed it up.
Okay. So you want me to take it from there?
Controlling. Professor Tree Fun, shot 1, take 2.
Sorry, what was that?
New generation.
Oh, sorry, you want to keep going. Okay, right. So what we needed was, the next generation of intravital microscopes. And we were really grateful that the ACRF took the leap of faith with us and trusted us to be able to build those microscopes and execute a vision in which we made those microscopes available not just to all the researchers at Garvan Institute, but throughout Australia. And what's been really exciting is that that's now allowed us to create an Australian intravital microscope community where we're able to share ideas, resources, and really help each other accelerate our research and head towards that final goal of truly understanding and being able to manipulate the immune system to help fight cancer.
And you can show us through one of these magical microscopes, can you?
We sure can.
Is there anything you want to say in general about medicine research?
Not really.
Covered it, do you think?
Don't know. I'm just answering questions as best I can.
Do you have anything, Steve?
It's very interesting to hear about the proteins that you're looking at specifically because they give rise to these cells, these B cells
Oh, there's right.
that target the particular pathogens that are attacking the body. Where do you think that technology could actually end up? I mean, to discover how you could even make those sorts of cells in immune systems that were weakened to create a
Right.
more efficient fighting machine against
Yeah.
disease?
Yeah.
If you just look at me when you answer.
Okay, all right. So, as an example of the sort of work that we do,
Take your time, it's fine.
okay, all right. So for B cells to differentiate, to become these plasma cells that secrete antibodies, they need to go to school. They need to learn, how to recognize and how to make the best antibodies to neutralize these viruses. And that school is what we call a germinal center. And so, when you have a vaccine and your arm, pits,
I'm so sorry. Oh,
do you need to see a doctor?
no, I don't think so. It's normally him.
Stuart doesn't trust doctors.
Yeah, I'm figuring that out.
last time I saw a doctor, they said there was no God. Anyway.
There's not
Vaccines.
one
Vaccines.
God. Or
Yes.
So these schools are called germinal centers. And so if you think about it, when you have a vaccine, sometimes your armpit swells up and it's painful. That's the lymph gland. That's where these germinal centers are. And what's really incredible is that they were first described way back in 1885, and yet there's still so many things we really don't know about them. And one of the reasons for that is because these are very dynamic, living structures that form and then disappear. When they're needed, they're there. When they're no longer needed, they go away. And so the intravital microscope allows us to get inside these structures, inside these germinal centers, to see these interactions, particularly between these B cells and the other cells that might be giving them instructions in terms of what to do and how to do it. And so we are now beginning to understand some of the rules of behavior that determines, the fate of the B cells, whether it becomes a plasma cell or not. Our hope is that that knowledge then would allow us to be able to engineer better vaccines, not just vaccines against, say, viruses, but vaccines against things that have been really, really hard to develop for many decades. We still are faced with a global burden of disease against things like tuberculosis, malaria, HIV. So these fundamental knowledge gains that we're making, we're hoping will be able to be translated into strategies that can make better vaccines against these difficult targets. This knowledge is also really important because, as it turns out, in a lot of cancers, the cancers where the immunotherapies like the checkpoint inhibitors are very effective, those are cancers in which you can find these schools, these germinal centers, sitting not in the lymph nodes, not in the lymph glands, but actually sometimes in the cancers themselves. So the question then becomes, can we engineer these structures in people with cancer to help make the immunotherapies more effective? Because while it's been amazing that the checkpoint inhibitors have had such a tremendous impact on cancers, they don't work in all cancers and they don't all work in every patient with a cancer. And so we'd like to be able to, move towards a situation where we can take this knowledge to improve the efficacy of these inhibitors in all patients.
Not a good answer.
Is that what you wanted?
Yeah, I think so.
Look, it's really interesting, because of course, I asked the question, how is it that some individuals' immune systems
are different,
being able and so different and being able to actually isolate and attack these things really quickly and kill them, stone dead, and how is it that some people just don't?
Yeah.
And then there's the idea about immune systems becoming exhausted after an exposure to a really long illness.
Yeah.
But it's just interesting. Do you think we're at the end of kind of, understanding the immune system and how it works in humans?
I think we're only at the beginning of that journey. I think increasingly the tools are now becoming available for us to pull apart all these processes. I've talked a lot about seeing these behaviors, seeing what it is that the cells are doing inside our body to help fight infection, to fight cancers. The next step then is also then to understand why are they doing those behaviors? What are the rules that are determining those behaviors? And then ultimately then, what are the molecular cues that they're taking? Because those molecules then become drug targets that we can then use to change the outcome of a cell's behavior, which then leads to a change in the outcome of whether a cancer grows or gets eliminated.
What's a drug target?
Sorry?
What's a drug target?
Oh, like, when you make a drug, it will target a molecule, like...
Because pathogens are normally molecules?
Because pathogens have molecules on their surface. They're decorated on this, it's the equivalent of, I don't know, like, how do you tell an echidna from I don't know, a koala bear, you know, it's like where it is, you know, in a tree, on the ground, but also echidnas have spiky things sticking out. So those spiky things are the targets,
And
right?
they're a defence also for the actual.
Yeah. So like, for example, COVID caused by the SARS-CoV-2 virus, they have spike proteins, right? So that spike protein becomes the target. And similar, cancer cells will have things on their cell surface that they need, right? Because it's important for their survival, right? Because it provides the signals that they need. So you can target that.
That's interesting.
Right.
So what does a bad guy look like? It's a molecule that has proteins on the surface, and that's what gives it its.
Well,
distinct character.
every living thing basically has a protein on its surface. The difference between a bad guy and a good guy might not be just the proteins, but whether that protein's mutated. So like in a cancer cell, for example, it might have a mutation that allows it to express a different protein that signals more. So instead of being told to stop growing, it's telling itself, oh, keep growing, keep growing. So, but because it's mutated now, it's different, our immune system should recognize that as dangerous and then destroy it.
Right, I think that's all good. I mean, you can now go into telomeres and it's all that stuff we've covered.
Yeah, but telomeres are the ends of the chromosomes.
That's why they keep growing.
Yeah, yeah. Yeah, but it's interesting because the proteins give off signals, and this is what the immune system.
It can.
So this is a way that the immune system detects
Yeah.
pathogens.
Yeah, one of the ways, yeah.
One of the ways.
Yeah.
Oh, right, okay. One of the ways. There's more.
There are many ways,
There's
yeah.
more.
There are redundancies, yeah.
Wait, there's more.
Wait, there's more.
Okay,
Yeah, I think so.
we're done then?
I think you get to show us the microscope.
Oh,
on
right,
stage,
okay.
Not necessarily now.
Have that
Look at that.
cutting?
We good?
Yeah.
All right, okay, and all right, and when you're ready, Tree, thank you.
So you just want to explain what's going on, or?
I'd like to look into the periscope first.
Oh, down here?
Yeah.
Like I was before?
Okay, yeah. And just go out and come in again.
No, you've got the cough. What's going on?
Okay, and we'll just stand you out, and then come in again like that. Oh my God. Sorry. All right, once more. We'll try again. And okay, and then we'll go out again there, and in again.
Oh, wow.
Well, we'll come in and be a bit lower when you come in.
Oh, like that?
Yeah, and that'll looks more like looking in. And action. We'll try more. Is this a little bit soft, please?
Oh, slower, you mean?
And action. Now we'll get you just to remember how we were turning your face to a slightly.
Oh, yeah, right.
Yeah, okay. And we'll try that again. And action. Okay, that's that one. Good. Okay, now we'll do one where, in fact, we do that again. Wait for it, and action. Sorry, I'll try one more again and frame it better. And
Okay.
action. And one more. And out again. Cut that. Right, and slap.
I haven't corrected.
Is it clap?
Clap.
One.
Mark it.
And close up.
Alright, and mark, roll up, marking.
Professor Tree Fun. Shot 3, take 1.
Start on the okay, so we'll transition across. So start on the machine. Felix.
Copy.
Alright, and
Copy.
action tree.
So what we're looking at here, do you want me to look at the camera or to the screen?
A bit of both.
Okay, all right.
And wait 1 second, and I'll say, and action tree.
So the two-photon microscope is incredibly powerful because what it allows us to do is to get deep inside living tissue in a live animal. So what you're seeing here is the lymph node in a mouse, and it's been immunized. So we're looking at the B cells making an immune response to the vaccine that we've given the mouse. So just to orientate you, this is the capsule of the lymph node. So that's the outside, and this is the inside. And this gap here, this space, is where the lymph flows in from the site where we've immunized. And so if you can imagine, if there was a virus or an antigen coming in, that's what will be captured by these cells, these macrophages in red. And you can see they've got all these little arms that are waving around. And what they're doing is they're sampling the lymph, trying to see what's coming in and capturing it. And what they would then do is hold it and then present it to these green cells, these B cells. And it's the job of these B cells to then find the vaccine antigen that's been captured on these macrophages and react against that. And as you can see, the cells are highly motile, they're very dynamic, they're running around the entire B cell follicle and talking to these other cells. And that's the sort of thing that we can see with the microscope that we wouldn't otherwise be able to do and see, which is these dynamic cellular interactions in real time, essentially getting under the hood and seeing what's happening in an immune response when we have a vaccine. Was that a minute?
Just picking up that last little bit.
Oh, okay. Which last bit?
Just about the, that's what happens in the vaccine.
Okay.
Just the last little bit of waving around with the arms and that. Yeah.
Oh, it's waving too much?
No, it's good. That looks great. Okay, and when you're ready?
So basically, this here is giving us a window into what's happening inside our body when we have a vaccine.
Okay, good. All right, now we'll just cut there.
Cutting there.
And
Professor Tree Fan, shot for take one.
good. And set.
So what we're looking at here is the lymph node in a mouse that's been immunized. And you can see the capsule of the lymph node, that's the covering of the node. So this is the outside and this is the inside. And this black space here, that's the sinus where the lymph fluid drains into the node, carrying the contents of that vaccine that's been given. And these red cells here, with all their processes or arms waving around in that sinus, they're sampling the lymph, trying to see what's coming in, capturing it, and what they're going to do is then they're going to show it to these green cells, which are the B cells. And they're the cells that are going to become activated and become the plasma cells that make antibodies that's going to help neutralize the virus that we've been vaccinated against. And as you can see, these green cells, they're not just sitting still doing nothing. They're running around the lymph node follicle, talking to each other, talking to other cells, these red cells, which are the macrophages. They're talking to these blue cells, which are the T cells, and they're receiving signals constantly that tells them where to go and what to do. And so basically, we are looking under the bonnet of the immune response and seeing it in real time.
Excellent. Ready, Andrew? Rolling up.
Yeah, sorry, give me a second.
Okay.
We'll just tell. Sorry, I don't want to block live.
No, you're right. No, you want to put me like we are there. Let me turn this one around to you.
No, that's fine. You got it.
That's it. All right, and rolling.
Rolling.
And rolling, Andrew. Very good. Okay, end slide. And action, train. Good. And we'll do that again. When you're ready, Felix.
Do you want all the way to the screen or to in at the screen?
Into the screen, I think. And
So,
thanks, Trey. Action. End slide.
filter.
Thank you.
holding. Thank you, everybody. Tree fun 5, take 1. Cutting there.
And you want Felix mark it?
Yeah, sorry. In a funny position. That's. You're getting that.
Yeah, I can see it's all good. Okay,
Tree
mark.
fun. Shot 6, take 1.
Okay, and wait for it. And when you're ready, Felix.
Yeah, coming up. Sorry.
There you go.
Not moving parts.
Yeah.
Okay, so
Yes. Yes, he gets over
All right,
himself.
and thanks, Tree. Action. Okay, and we'll get you now to do again your explanation of all of that.
Oh, I'm going to explain it again
Yeah.
while I'm standing here?
Yeah.
Okay.
Just look to the camera when you do it. If you can stand a little closer to them. Yeah, that's great. Perfect. Right in there. That's great.
Okay.
Yeah, good. Okay, and when you're ready.
So what we're looking at here is the lymph node of a mouse that's been immunized. And so this is the capsule, the covering of the outside of the lymph node. And so on this side is the outside, and in here is the inside of the lymph node. And in blue, the capsule, just beneath it, this black space is the sinus where the lymph fluid carrying the contents of that vaccine enters the lymph node. And you can see these red cells with their little arms waving in the sinus. What they are doing is they're sampling the sinus. So these are macrophages, and they're trying to collect the fluid from the fluid that's coming in, the vaccine antigen. And then what they'll do is they'll hold it and they'll use it to activate these B cells, these green cells, which you can see here running around the B cell follicle. And so normally the B cell follicle contains these B cells that are running around trying to find these antigen, the contents of the vaccine that are meant to react against. And what they're doing, you can see, is they're talking to all these other cells, communicating, and then from that, these B cells become activated, differentiate, or become plasma cells to make antibodies. So essentially, we are getting under the hood of the immune response and seeing what's happening in real time.
No, that's great. Could you do just the last bit after it's all waving around and talk about how the B cells do it, but play it into the camera as opposed to just looking at the screen?
So the last bit about how...
Yeah, like from the sinuses where the fluid comes into the sinus and after the
Right.
in that black space.
But look at this camera, or...
Yeah, just to sort of like, you can
Okay.
refer to the screen and point
Okay.
it, but then do most of the talking into the camera.
Okay, right.
Okay, and when you're ready.
So what we're looking at here is the lymph node, and this is where the immune response is being made. So when you get immunized, the lymph node is where the vaccine antigen drains to and activates the B cells. So here you can see in blue the capsule or the covering of the lymph node, and sitting beneath that, this black space, that's the lymph sinus where all the fluid carrying the vaccine antigen comes into the lymph node. These red cells lining the floor are macrophages, and you can see their little arms waving around, and what they're doing is sampling the lymph that's coming in, seeing if there's anything dangerous that the immune response should make a reaction against. And if they see that, they'll capture it, and then they'll present it to these green cells, which are the B cells running around inside the follicle. And these B cells are trying to collect information to tell them what to do. If they get the right information, they'll become plasma cells to make antibodies. That's the purpose of the vaccine. And so these B cells, you can see, are very dynamic. They don't just sit still in one place. They're running around, communicating, talking to as many cells as possible. And if they get the right signals, they'll make the antibodies that you need to neutralize the virus that they've been vaccinated against. And so what we're looking at here then is essentially under the bonnet of the immune response.